Welcome to In-Silico Trials Real Impact, the In-Silico UK podcast that's revolutionizing the way we think about medical innovation. I'm Professor Alejandro Frangi from the University of Manchester, and I'll be your guide through this fascinating world of in-silico technologies. In each episode, we'll dive into cutting-edge research and groundbreaking news that are shaping the future of medical product development and regulation. From computational modeling to simulated clinical trials, we are exploring how digital approaches are transforming healthcare, making it safer, more efficient, more equitable, and incredibly precise. So, whether you're a researcher, regulator, a member of the public, or a patient representative, or simply curious about the future of medicine, tune in as we unravel the complex world of in-silico methods and their real-world impact. Get ready to discover how computer-simulated trials are leading to very real breakthroughs in patient care. In-Silico Trials, Real Impact, where algorithms and data are saving lives. Let's begin.
Welcome to the Deep Dive, where we cut through the noise to bring you the essential knowledge on important topics. We do the sifting so you don't have to.
And today, we're diving into something really specific, but pretty crucial for folks with insulin-dependent diabetes. It's about switching glucose sensors with the Medtronic 780G pump system.
Okay, yeah, let's get straight to it. We're talking about moving from the Medtronic Guardian Sensor 3, that's the GS3, which needs regular fingerstick calibration.
Right, to the newer one, the Medtronic Guardian 4 sensor, the G4S. And the big deal here is that the G4S doesn't need those calibrations. Potentially a lot easier.
Definitely simpler. Now, it's worth pointing out the study we're looking at happened when the G4S was still, waiting for the green light in the US, but it was already out there in other places.
Exactly. And our source material for this deep dive is a scientific paper. It's called In-Silico Evaluation of the Medtronic 780G System while using the GS3 and its calibration-free successor, the G4S sensor.
In-Silico.
Yeah. And what's interesting is how it pulls together different kinds of evidence, computer simulations, data from a clinical study, and even real-world commercial data.
Right. So let's start with that in silico bit. Computer simulations. Why are these becoming, such a big deal in developing diabetes treatments?
Well, it's really about creating what they call virtual patients. Think of them as sophisticated mathematical models running on a computer.
Okay.
They mimic how a person's blood sugar responds to things like insulin and food.
So like a digital test dummy, but way more advanced to try things out.
Exactly. And it's not just guesswork. These models are built on, decades of research into glucose metabolism. The paper mentions some well-known ones. The minimal model, the Uva-Padova simulator.
Familiar names in the field.
Right. But the Medtronic model used here is, it's built on a massive data set.
Oh, yeah. What makes it so big?
They used real-life data from Medtronic's CareLink platform. We're talking info from over 5,800 people who were using the previous pump, the 670G.
Wow, 5,800. So these virtual patients aren't just generic, they reflect real people.
Very much so. They capture a huge amount of variability. Things like how sensitive someone is to insulin, how fast they absorb meals.
Different ages, body types.
Exactly. Mm-hmm. The ages range from 1-year-old up to 88. BMI, median was around 26 based on over 4,000 of them. Gender was pretty evenly split.
Okay.
And even total daily insulin use, anywhere from 4 units a day to, believe it or not, over 200.
Wow.
Plus, they built in things like daily rhythms, circadian variations in insulin sensitivity, and even, real-life hiccups, like messing up doses sometimes.
They even factored in human error. Okay, so you have this incredibly detailed virtual population. How do they actually run the simulation to test the sensors?
So this is pretty neat. The Medtronic simulation environment actually runs the exact same software code that's on the real 780G pump.
Okay.
Yeah. And they connect this pump software to the virtual patient model. The software sees the virtual patient's glucose, decides on insulin doses, delivers them virtually, and the model calculates the glucose response.
So it's like a full dress rehearsal inside the computer.
Pretty much. It lets them predict glucose outcomes, check things like alarms, all in this controlled digital space.
That is clever. And the big advantage here, for this study anyway, was isolating just the sensor change, right? How'd they do that?
That's where Silico really shines. They took the same virtual patients with their existing 90 days of data.
Okay.
And the only thing they changed in the simulation setup was the sensor model. Run it once with the virtual GS3, run it again with the virtual G4S.
Got it. So any difference you see should theoretically be down to the sensor itself.
Exactly. It isolates that one variable.
Okay, but you can't just tell the computer use GS3, right? They had to build digital versions of these sensors. How'd they manage that?
Right. Good point. They developed what they call In-Silico sensor error models, basically digital blueprints of how each sensor behaves.
Based on real sensors?
Yeah, based on loads of real-world data from actual GS3 and G4S sensors. They used pretty advanced data analysis and modeling techniques.
So they're building in the typical noise, the inaccuracies you see with real sensors?
Precisely. They modeled the random fluctuations, the potential for errors. They even considered how accuracy might change over the sensor's lifespan. The aim was to make the virtual sensors behave like the real deal, warts and all.
Okay, makes sense. And did they check if these digital sensors were, realistic enough?
They did. They compared the accuracy of the In-Silico sensors against real-world data, looking at performance above and below that 70 milli-GDL threshold. The paper says there were some, minor differences, but nothing they thought would be medically significant. Nothing that would really change how the pump system worked in the simulation compared to real life.
All right, so we've got the virtual patients, the digital sensors, the simulation setup. Let's get to the results. What happened when they simulated that switch from GS3 to G4S? What did the different data sources show?
Okay, let's start with the clinical study data. This was from the Continued Access Study, or CAS people who'd been in the original 780G trial. 2 age groups, 7, 17 years, and 18 plus.
Got it. What changed when they moved to the G4S?
Well, average glucose went up slightly, about 2.1 milli-GDL in the younger group and 4.8 milli-GDL in the adults.
Okay. What about lows? Time below 54 milli-GDL.
That actually went down a bit, decreased by 0.2% in the kids and 0.4% in the adults. Same direction for time below 70, small decreases there too.
Interesting. So slightly higher average, but maybe less hypoglycemia risk. What about time in the target range, 7180?
That was mixed. For the 717 group, time in range actually increased slightly by 0.8%. But for the adults, it went down just a tiny bit, 0.4%. And time above 180 was kind of the flip side. Small decrease for kids, small increase for adults. Time way up above 250, increased slightly for both.
And they mentioned pump settings.
Yeah, most participants were apparently using a target of 100 milli-GDL and an active insulin time, the AIT of 2 or 3 hours.
Okay, that's a clinical study snapshot. Now, the European commercial data, this is more real world, everyday use, right? Bigger group.
Yes, 1,335 users in Europe who made the switch. No exact ages, but the pump settings were varied. About half used a 100 target, 30% used 110, 20% used 120. AIT was split between 2 hours and 2-3 hours.
More diverse setup. And what did that data show?
Average glucose increase was a bit higher here at 5.2 milli-GDL.
Okay.
But similar to the clinical study, time below 54 and time below 70 both decreased slightly. Reductions of 0.4% and 0.6%.
So that pattern holds. Less time low.
Seems so. Time in range, though, decreased by 1.1%, and time above 180 went up by 1.8%, time above 250 up by 0.8%.
Okay, so the commercial data seems broadly similar to the adult group in the clinical study. Average glucose up, lows down, time in range down a bit, highs up a bit.
That's a good summary. Now, let's bring in the simulations. What did the computers predict? They tested different settings, right?
Yeah, targets of 100 and 120, AITs of 2 and 3 hours, and the 2 age groups.
And the simulations generally predicted that average glucose would increase with the G4S switch. The range was about 2.2 up to 4.5 milli-GDL increase, depending on the settings.
So right in the ballpark of what the real-world data showed.
Pretty much, and for lows.
Also matched the trend. Simulations predicted either no change or a slight decrease in time below 54. Max decrease was 0.2%. Time below 70 also predicted to decrease slightly, maybe up to 0.5%.
Okay, so consistent on the lows. What about time in range and highs? Simulations generally predicted a slight decrease in time in the 70-180 range, somewhere between 0.9 and 1.5%.
Matching the commercial data and the adult clinical data again.
Right. And consequently, time above 180 was predicted to increase by 1.0% to 1.9%. Time above 250 also predicted to increase a bit, 0.2% to 0.7%.
Okay, wow. So when you put the simulation results side by side with that large European commercial data set, how well did they line up overall?
This is actually one of the most striking findings. The agreement in the direction of change between the simulations and the European commercial data was, well, perfect.
Perfect.
Yeah. For average glucose, time below 54, time below 70, time in range, time above 180, time above 250, every single metric moved in the same direction in both the simulation predictions and the real-world commercial observations.
That's pretty impressive validation for the simulation model, isn't it?
It really is. It suggests the model is doing a remarkably good job of capturing the essential dynamics of the system and the sensor change, at least directionally, compared to large-scale real-world use.
But you mentioned earlier the clinical study data wasn't quite a perfect match, especially for the kids.
That's right. The CIP 321 clinical data, specifically for the 7-17-year-olds, showed an opposite trend for time and range. It went up slightly in the study, whereas simulation and commercial data showed it going down. And similarly, time above 180 went down slightly for those kids in the clinical study, while the other data sources showed it going up.
Any thoughts on why that younger group in the clinical study might have looked different?
Well, the paper mentions a few possibilities. The clinical study had fewer participants, obviously, and critically, the group using the G4S in the continued access phase wasn't necessarily the exact same cohort under the exact same conditions as those who used the GS3 in the original pivotal trial.
Ah, okay. So maybe other factors changed for those kids between the two phases.
It's possible. Real life, even in a study, is messy. There could have been other shifts in their management or behavior that influenced those specific outcomes, especially in a smaller group.
Right. So we're talking about changes like 0.2%, 1%, maybe 1.5% in some of these time metrics. How do we think about whether that's actually meaningful, clinically significant?
That's the million-dollar question, isn't it? The paper points out something useful. A 1% difference in time works out to about 14.4 minutes per day.
Okay, so a 1% drop in time and range is about 15 fewer minutes in target each day.
Exactly. So these small percentages aren't necessarily trivial when you add them up. However, the researchers also looked at the difference in the size of the change between the simulation predictions and the real-world commercial data. The biggest disagreement there was only about 0.3% or 0.4%.
Which translates to
maybe 5 or 6 minutes per day. And the argument is, for most people, a difference of 5 or 6 minutes in one of these categories probably isn't going to feel like a major clinical shift.
Fair point. So the predictions and reality were directionally aligned, and the magnitudes were pretty close, likely within the range of, day-to-day noise for many users.
That seems to be the takeaway, yeah.
And why might we see even those small differences between simulation and real life? You mentioned real-world adjustments.
Yeah, that's one potential factor. The simulations model the algorithm and the physiology, but maybe not always the subtle ways people adapt their behavior.
Like
if someone using the G4S notices their average is running just a touch higher, maybe they make a tiny tweak to their basal rate or how they count carbs for a specific meal. Those real-time human-in-the-loop adjustments.
Things the simulation wouldn't necessarily capture.
Exactly.
Yeah.
The simulation follows its programming based on the virtual patient, but real people might fine-tune things based on their lived experience with the system.
Okay, so let's try and wrap this up. What's the bottom line from this deep dive, the main implications?
Well, I think the first big takeaway is that this study really shows the power of Medtronic's simulation platform. It could predict the outcomes for a next-gen sensor pretty accurately, which is quite impressive.
Gives confidence in that modeling approach.
Definitely. And for users of the 780G, the core message is that switching from the older calibration-requiring GS3 to the newer calibration-free G4S seems safe.
No major red flags in terms of clinical risk.
Right. The data from simulation, clinical trials, and real-world use all suggest that while there might be some minor shifts in glucose metrics, maybe a slightly higher average, slightly less time low, the overall clinical outcomes are generally maintained.
That's reassuring, especially given the convenience benefit of not needing calibrations with the G4S, and it really highlights the value of these in-silico studies, doesn't it?
Absolutely. They can augment clinical trials, maybe even replace some smaller ones. And that ability to isolate one single change, just the sensor, in this case, is something that's incredibly hard to do perfectly in a traditional trial.
You can test so many scenarios, so many virtual people.
Exactly. Far more than you could ever recruit for a standard study.
Of course, neither approach is perfect on its own, right? Clinical trials have limitations.
Sure, sample size, diversity sometimes, controlling real-world variables.
And simulations are still, at the end of the day, models, simplifications of complex biology.
True. They're incredibly sophisticated simplifications, but still simplifications. They work best when validated against real-world data, like in this study.
So the overall picture, moving from GS3 to G4S on the 780G, looks like a reasonable switch, supported by both computer predictions and real-world findings, with probably minor, clinically insignificant changes for most.
That sums it up nicely. The convenience gain seems to come without a significant clinical penalty based on this evidence.
Well, this has been fascinating, really peeling back the layers on how this technology is evaluated using these pretty advanced virtual methods.
It really is cutting-edge stuff. And it leaves us with a thought, maybe for you, our listener, to chew on.
Go on.
As these virtual patient models and in-silico trials get even better, how might that change the whole game for developing and approving new medical devices, new therapies? Could we get more personalized treatments faster? Maybe reduce reliance on some traditional, expensive, sometimes slow clinical trials.
Yeah, where else could this kind of super detailed modeling make a difference in health? It's definitely something to think about.
Food for thought.
Absolutely. Well, thanks for joining us for this deep dive. We'll catch you on the next one.